Good afternoon, everybody, and welcome. My name is Vipul Jareth. I'm the CMO at Alimentiv and a gastroenterologist in Canada. I'd like to thank you all for joining our live stream event today, which is entitled Real-World Evidence in Drug Development for IBD. Almost 500 people have joined this from various parts of the world, and we're grateful for you for spending your time with us today. We've had a number of these events, as you'll know, over the last year, and they've focused on other areas in IBD, such as precision medicine, fibrostenosis and Crohn's disease, and histopathology, just to name a few. We've got 4 fantastic speakers today, and I'm shortly going to be introducing the chair for the event, Dr. Jessica Allegretti. Just as a reminder, this event's a bit longer than usual. They're normally 60 minutes. This is going to be 80 minutes. So it's starting at 1 p.m. Eastern time and ending at 2:20 Eastern time. And there's going to be about a 15-minute Q&A session at the end as well, which can address some of the questions. Just a few housekeeping things. If you've got any technical issues, please use the questionnaire on the right-hand side of your screen, and someone will respond to that. For optimal viewing, try and close programs such as Teams. And as I mentioned, there will be Q&A at the end of this. So if you want to submit any questions on the right-hand side of your screen, there's an area that you can do that. We won't be able to answer everything, but we'll do our very best to answer as many things as possible. And if you have any issues viewing the screen, please click the stream option button 2 on the bottom right of your screen. So with that, I'd like to welcome the chair for the event, who is Dr. Jessica Allegretti. It's a real pleasure to have Jessica here doing this, who will be introducing the speakers, but also moderating all the Q&A as well at the end. Dr. Allegretti is a gastroenterologist at Brigham and Women's Hospital in Boston. She's the director of clinical research there. She's associate director of the Crohn's and Colitis Center, and she pioneered the fecal microbiota transplant program there and has become really recognized as one of the world's authorities in that area. Her lab, in addition to the microbiome, focuses on novel therapeutics for IBD and runs multiple investigator and industry-sponsored studies there. So with that, Jessica, I'd like to thank you for chairing this event and hand over to you. Thank you.
Thank you so much. It's great to be here today. I'm really looking forward to the presentations and the discussion. And it is my pleasure to introduce our first speaker, Dr. Martin Ladouceur, who's a methodologist specializing in health research with over 15 years of applied experience in clinical research and consulting. He is currently the VP of Real-World Evidence at Alimentiv, where he leads and oversees the growth, scientific service offerings, and consulting of the newly formed Real-World Evidence Unit. As a subject matter expert in real-world evidence methodology, he focuses on bringing scientific evidence to clients, fit-for-purpose study design, and the use of real-world evidence. Hand it over to you, Martin.
Good afternoon, everyone. My name is Martin Ladouceur. I'm the Vice President of Real World Evidence here at Alimentiv, and it's a real pleasure to be here today to give you a brief overview of real world evidence from a regulatory standpoint with some inputs on external control arm. So the recent pandemic had important impact on clinical research, where nearly 80% of non-COVID trials were stopped or interrupted due to the COVID pandemic. The majority of current trials disruptions are due to slow enrollment, significantly impacting trials in indications for which clinical trial recruitment is already challenging. The recruitment impact across indications has highlighted the need and potential advantages of innovative study design. As a complement to classic randomized clinical trial or external control arm, which could help to mitigate recruitment challenges and create efficiencies in the drug development pathways. Okay. First things first, can you see, just one second. Okay, first thing first, what do we mean by external control arm? It's a design that compares a group of patients receiving the test treatment in an interventional study to a control group of patients external to the study. In other words, a control group of patients from a different population. Control can arise from either historical, patient treated at an earlier time, or contemporaneous patient treated during the same period but in another setting. The use of an external control arm can confer important benefits to patient and industry and reduce patient and site burden. A key advantage of using an external control arm is that the patients can receive a promising therapy. As well, external control arms significantly improve trial efficiency and lower the cost and time needed to conduct clinical trials. Real-world evidence are no longer limited to phase 4, but are now seen at all stages of drug development to create efficiency. So this figure gives a visual representation of the study design. External control data can be extracted from many data sources and is filtered, for example, based on patient characteristics, trial inclusion, exclusion criteria, and matched to trial data through propensity score matching, exact matching, IPW, or dynamic borrowing, for example. If an external control arm is used to support a regulatory submission, adequate data based on predetermined patient selection criteria and pre-specified statistical analysis plans are required. One important aspect to keep in mind is that there are many ways to design a study in which an external control arm can be leveraged. It could be to add value to an already ongoing single arm study. So you have your single arm study that is running and you're adding the comparator arm after. You could be designing at the beginning where you have your treated patient in an interventional group and untreated in the non-interventional setting right at the beginning of your study design, or a mix of those two ideas where you could have a randomized study with a limited number of untreated patients and you complement the number through external control. So chances of success increase if the standard of care or control group are well established. The 21st Century Cures Act of 2016 required that the FDA develop a program for evaluating the potential use of real-world evidence in regulatory submissions, including a framework, guidance, demonstration projects, and stakeholder engagement. What is the FDA looking for when it gets to the use of real-world data? Well, three main components: design, data, and requirement. The FDA will consider whether the real-world data are fit for use, whether the trial or study design used to generate real-world data can provide adequate scientific evidence to answer the regulatory questions, and whether the study conduct met FDA regulatory requirements, for example, informed consent, study monitoring, and oversight, and data collection. Real-world data comes with challenges, obviously, such as inconsistent format of real-world data sources, global and regional variation in data standard, as well as standard of care difference across region, different methods used to aggregate data, and importantly, overall quality of data. To mitigate the challenges of implementing FDA data standard to real-world data, FDA recommends that sponsors document the processes used to curate, manage, and transform real-world data, as well as document any difficulties encountered. So transparency at its best. Seek early discussions with the appropriate FDA review division for further guidance on any planned submissions containing study data derived from real-world data. And leverage pre-submission processes to ensure alignment with the FDA. So how can we integrate an external control arm design into the FDA guidelines? Well, the key is that the data must be fit for purpose and that there is no perfect database to meet all external comparators' need. The right data must be chosen to address the particular research question at hand. Remember, it's easy to do a bad external control arm, and there is no one size fits all. The data should come last, in a sense that what is needed first is why the external control arm should be put in place. Then design the best study, taking into consideration the feasibility to collect the appropriate outcome. Everyone have access to real world data, and they are common. But the real expertise is about designing a fit for purpose study design that will demonstrate the value of that real world data into clear, convincing, and meaningful evidence that will support your research. If you want to be convincing, you need to strategically think about how you're going to design the study, what data will contribute to the evidence, and how those will maximize the acceptability by relevant stakeholders. So the big question is, are external control arm accepted by authorities, and are they accepted for conditions outside oncology? Historically, ECA have been used in oncology and rare diseases. Between 1999 and 2014, 60 external control arms were accepted by the FDA and 44 by EMA. And only a handful of them were in non-oncology. But you can see there's a slight change from the last 15 years to the last 4 years. The numbers have really exploded in the non-oncology setting. There's some discrepancy between the FDA and EMA approval. The FDA extensively uses an accelerated approval process, results based on a surrogate endpoint with confirmatory RCT conducted subsequently, while the EMA does not frequently use its equivalent process, conditional approval. So companies typically engage with the FDA throughout the drug development process, and companies do not necessarily interact with EMA during drug development. So there is a huge momentum in the industry right now, be it among big pharma, biotech, and consulting company around the use and design of external control arm. ECA won't certainly become the norm for all indications in the near future, but when there are exceptions to the norm, as we just saw, this creates a momentum and interest to increase the context in which such studies would be allowed. An interesting question is whether indication facing recruitment challenges, such as IBD, EOE, and NASH, would be the next one to benefit from such study design. To get there, what we need are proof of concept. Validation studies will be needed for indication with no prior studies to serve as a reference. Further, FDA guidance on the use of ECA will support clinical development and products approval. In a medium term, we can expect to see more and more of those submissions, including using external control arm. So we can see that becoming more regularly used seems plausible. But in the long term, with RCT be replaced with external control arm, I think that's a long shot. We don't think this necessarily the field will move to completely replacing them, but you'll see more and more of those applications. So innovative design are needed to face the challenges of tomorrow's research. External control arm are one of them. ECA, and like any novel approach, it's important that people do these things and share their learning. ECA are attractive and could be very powerful tools to complement clinical trials, but as you all know, with great power comes great responsibility. So thank you very much, everyone.
Perfect. Well, thank you so much, Martin. That was excellent. And now we're going to be hearing from Dr. Niels van Casteel, who is trained as a clinical pharmacologist and holds a bachelor in pharmaceutical sciences and a master in drug design and development from the University of Leuven in Belgium. He is currently the president of Acela Bio, where he oversees and directs all business and scientific aspects. In addition, he is an associate professor of medicine at the University of California in San Diego, and his main research focus is on the application of clinical pharmacology and translational medicine to ascertain efficient drug development. Over to you, Niels.
Thank you so much, Jessica, for the kind introduction, and thanks for the opportunity to present here today. So I'll be talking about a case study on vedolizumab to use real-world evidence to inform pharmacokinetic modeling for IBD therapeutics. So before getting into vedolizumab specifically, I would like to take a step back and provide some general background about the first monoclonal antibodies that became available for the treatment of inflammatory bowel disease, which are, I'm sure, well known by the audience, and these are the tumor necrosis factor antagonists. And so what we know about the TNF antagonists, that about a third of patients do not respond to these treatments, and of those patients initially responding, there is a proportion of patients that lose response over time. And we found out that that may be attributed to suboptimal drug exposure, and therefore therapeutic drug monitoring with measurement of drug concentrations is used to optimize dosing and treatment outcomes in patients experiencing non-response or loss of response to TNF antagonists. So although the role of TDM is well characterized for that class of drugs, and it's actually taken up in guidelines internationally, it is not well defined for vedolizumab in the treatment of inflammatory bowel disease. So what do we know about therapeutic drug monitoring for vedolizumab? Well, a post-hoc analysis of the Gemini phase 3 registration trials showed a positive exposure-response relationship between serum vedolizumab concentrations and clinical outcomes. And although there is emerging data supporting the role for reactive therapeutic drug monitoring with vedolizumab, which would be a time of secondary loss of response, really additional evidence should be generated to support its implementation in daily clinical practice. So therefore, we designed a trial to assess basically the correlation between vedolizumab drug concentrations and therapeutic outcomes in a real-world setting. So we performed a multi-center observational cohort analysis of real-world clinical and model pharmacological data to evaluate the exposure-response relationship of vedolizumab in IBD patients. So our main study objective was to evaluate the relationship between vedolizumab exposure and response. In order to do so, we first needed to assess the clinical, endoscopic, and biological response rates to vedolizumab therapy. If we observed that there was a positive correlation between exposure and response, we also wanted to delineate drug concentration thresholds that are associated with clinically important outcomes. As well, we wanted to perform multivariable logistic regression analyses to understand if there were covariates at baseline that may be predictive of response to therapy. And finally, we also wanted to evaluate the safety of vedolizumab in this clinical practice cohort. So to provide you a bit of information on the study design, we basically performed a retrospective chart review where we pooled all of the data on real-world IBD patients in clinical practice that were treated at 9 academic expert research centers across 6 countries. So we collected patient demographics, treatment information, pharmacokinetic data, and treatment outcomes, and some leveraging existing databases. As Martin already alluded to, due to the retrospective nature of these types of studies, complexities owing to sparse and heterogeneous data were expected, with inter-site differences in treatment modalities, treatment outcomes, availability of therapeutic drug monitoring data, and also assessment time points. However, this also presents an opportunity to actually evaluate some of the inter-site variabilities, for example, in treatment modalities, and how those may impact the outcome to vedolizumab therapy. So what I would like to do next is to provide you some more detailed background about the methods that we applied for our eRelate study. So we collected data on patients that were treated with vedolizumab between a period of June 2014 and December 2019. We enrolled patients who had a confirmed diagnosis of ulcerative colitis or Crohn's disease and who had at a minimum age of 17 years or older. In particular, we wanted to enroll patients who had at least a follow-up duration of 1 year on vedolizumab treatments. We were able to enroll in total 695 subjects, of which 304 were ulcerative colitis and 391 were Crohn's disease patients. As well, in order to support population PK modeling, we required the following covariates to be available for those subjects: baseline body weight, baseline serum albumin, vedolizumab dosing history, and it was optional to also provide observed serum vedolizumab drug concentrations. And why those covariates were important was because we incorporated them into an existing population PK model that was developed based on the Gemini trials. And based on this population PK model, we were able to update for individual patients based on their individual covariates at the start of enrollment, as well as based on their individual vedolizumab dosing history, vedolizumab drug exposure levels throughout the entire observational phase, and to correlate that with the observed outcomes. So this is what you can see here on the far right, is that we were able to generate vedolizumab exposure levels, as well to correlate that with the observed outcomes. So you can see here at the bottom right that we were focusing on key outcome measures such as clinical remission, endoscopic remission, and deep remission, which was a combination of both clinical remission and endoscopic remission, or in the absence of endoscopic remission, we applied biological remission, which was defined as CRP less than 5 or fecal calprotectin less than 150. And we assessed those observed outcome measures both at the end of induction at week 14, as well as during maintenance therapy at week 26, and at the end of 1 year at week 52. So here you can see the baseline demographics of the cohort of 695 IBD patients. And for those of you who are familiar with the Gemini trials, this very much is in line with the baseline demographics of the clinical trial. However, what is difference here is that there was a proportion of patients who started vedolizumab therapy who had no active disease or mild active disease. And that is to be expected in a clinical practice setting where oftentimes patients are started on new therapy prophylactically. So before showing you the results on the exposure response analyses, I first wanted to provide some data on the method that we applied using Bayesian statistics to basically generate vedolizumab exposure levels in all patients throughout the entire follow-up. So you see here an XY plot with, on the Y-axis, the predicted vedolizumab concentrations using population PK model, and on the X-axis, the observed vedolizumab concentrations that were provided by some sites on some patients. And as you can see, there was a very good correlation as well as agreement between both measures. This graph shows the combined cohorts of ulcerative colitis and Crohn's disease patients, whereas here on the following graph, what you can see is that broken down for ulcerative colitis patients on the left and Crohn's disease patients on the right, essentially showing similar results. So overall, this demonstrates that incorporating the observed concentrations into population PK model using a Bayesian approach allows for pooling of data, even if there was variability between the assays used to measure vedolizumab concentrations across the different sites. So next, I would like to go over the clinically important outcomes that we observed in patients with ulcerative colitis and Crohn's disease. So here you can see the key outcomes for clinical remission, endoscopic remission, and deep remission for ulcerative colitis in teal and for Crohn's disease in yellow. What you can see is that approximately 40% of patients with ulcerative colitis or 35% with Crohn's disease were in clinical remission by week 52. Endoscopic remission was achieved by about 40% and 74% of patients with UC and Crohn's respectively by week 52. From a deep remission perspective, only about 20% of patients with both ulcerative colitis or Crohn's disease attained the outcome by week 52. So now that we have the exposure measures as well as the observed outcomes, we wanted to evaluate the exposure-response relationship. And what you can see here is the data of time concentration profiles of both ulcerative colitis and Crohn's disease patients combined. At the top, you can see the clinical remission outcome, and at the bottom, the deep remission outcome. On the left is a week 14 outcome, and on the right is a week 52 outcome. And what you can see on these graphs is for the remitters in red, they consistently had higher trough concentrations than the non-remitters in blue. In particular, you see a greater separation between remitters and non-remitters for the deep remission outcome, which is a more stringent outcome at the bottom, as well that the separation appeared to be greatest during the induction phase at week 6 and week 10. In blue, you can see some of the concentration thresholds highlighted that we were able to delineate that were associated with achievement of those outcomes. Besides looking at trough concentrations, we also were able to look at other measures of exposure because of the population PK modeling approach that we applied. What you can see here is a typical quartile analysis. For those of you who are less familiar, you basically rank patients on their exposure measure from low to high, divide them into 4 equal groups, and then look at the proportion of patients who achieve the outcome. What I've highlighted here on these 2 graphs is the deep remission outcome at week 14 on the left and week 52 on the right. If we start off on the right-hand side for both graphs, what you can see there are the quartile analyses for trough concentrations at week 6, week 14, and week 22. And in general setting, this is quite similar to the linear exposure-response relationship that was also observed in the post-hoc analysis of Gemini. What our new findings are the results in teal, which show the average concentration in the periods between week 0 and week 6, and the importance of that exposure measure in association with achieving the outcome. As well as on the far left, for both graphs, what we were able to observe for the first time is that actually clearance of the drug at baseline when patients start vedolizumab treatment is strongly associated with achieving the deep remission outcome at week 14 and week 52. In particular, those patients who have accelerated drug clearance are at risk of not achieving short and long-term outcomes with vedolizumab. Next, we wanted to look at the predictors of outcomes at baseline. And we observed that age at baseline, prior IBD surgery, use of concomitant immunosuppressants at the start of vedolizumab treatment, and or lower baseline vedolizumab clearance were associated with achieving clinical or deep remission at week 52. In particular, baseline vedolizumab clearance was also a significant predictor of endoscopic remission at week 14 in the UC group, biologic response at week 52, and biologic remission at week 52, thereby underscoring the importance of this characteristic in influencing treatment outcomes. Here you can see the findings from a safety aspect in this clinical practice cohort, and without going into too much detail, in general, these results were very reassuring and no additional safety signals were observed for vedolizumab for both Crohn's disease and ulcerative colitis. So in summary, I would like to share with you that this is the largest exposure response analysis of vedolizumab in clinical practice data to date. and provides novel insights about the association between pharmacokinetic parameters and short and long-term treatment outcomes. As well, the Bayesian updating of the population PK model showed strong correlation between predicted and observed drug concentrations and really allowed for robust exposure response analysis of real-world data. And it is our understanding that this approach could also be applied for other therapeutics and other indications. By using this approach, we were able to show that there's a positive relationship between exposure response, between serum vedolizumab concentrations and clinically important outcomes. In particular, vedolizumab clearance was found to be negatively associated with clinically important outcomes and appears to be an important predictor. And finally, the frequency and type of SAEs observed in the study are consistent with the known safety profile of vedolizumab. So in summary, our study provides real-world data demonstrating exposure-response relationship to vedolizumab, where lower vedolizumab clearance at baseline and higher drug exposure, especially early on in a treatment, are positively associated with better outcomes over the long term. Thank you very much for your attention.
What a fabulous talk. Next, it's my pleasure to introduce Dr. Michael Kappelman, who is a professor of pediatrics and epidemiology at the University of North Carolina in Chapel Hill. His primary research interests include patient-centered outcomes research and comparative effectiveness and safety research in the area of inflammatory bowel disease, as well as studies of the epidemiology, costs, and utilization of resources by children and adults with IBD. He has experience in both observational and interventional study designs and has received research funding from the NIH, AHRQ, CDC, PCORI, foundations, as well as industry. Over to you, Michael.
Thank you so much for the overly kind introduction, and it's my pleasure to talk with you all about comparative effectiveness research in IBD using real-world data. I have a number of disclosures on this slide. And over the next 20 minutes, what I hope to do is introduce you to the great need for comparative effectiveness research in IBD, discuss some of the differences between randomized controlled trials and real-world evidence and where real-world evidence fits into the research landscape. I'll give a number of examples of observational studies that generate real-world evidence. We'll talk about both some methodologic issues as well as examples of representative studies. And in the end, conclude with a discussion of pragmatic trials, a bit about the design philosophy for this study type, and a case study. And so there really is an urgent unmet need for comparative studies in inflammatory bowel disease. We have more treatment options than ever before and a robust and exciting pipeline with many promising new prospects. And as a result, when I trained, IBD care was easy. We talked about the therapeutic pyramid, where we start off with aminosalicylates and then work our way up to prednisone and budesonide and up the pyramid as shown here. And of course, now all of that is a bit past tense, and IBD care these days is much more complicated. We really do have a number of treatment challenges, including medication selection. Do we use immunomodulators before biologics, vice versa, or in combination? How do we think about positioning of anti-TNFs versus newer classes of biologics or small molecules? And it's actually even more complicated than drug A versus drug B, as if one was treating a acute streptococcal pharyngitis. We're dealing with a chronic long-term illness, and so really need to think about therapeutic strategies, the timing and sequencing of treatments over a patient's long journey with illness, how to consider issues related to escalation, de-escalation, and even dose optimization. And so we really are balancing many of the benefits and risks of therapies in an evidence vacuum. And so comparative effectiveness really is an urgent research need in the IBD space. In 2009, the Institute of Medicine issued a comparative effectiveness report that identified studies of biologics and IBD as a top quartile research priority. 2 years later, the Agency for Healthcare Research and Quality issued a paper that concluded that comparing Crohn's disease medications directly will help understand the effectiveness of medicines available for use in clinical practice. And I would postulate that in 2021, with the growing treatment armamentarium as it is, the need for comparative effectiveness research in IBD is greater now than ever before. And we're beginning to see a number of head-to-head studies emerge. On the left, the Varsity trial, which compared vedolizumab to adalimumab for moderate to severe ulcerative colitis. On the right, the CView data, which has been presented in abstract form, comparing ustekinumab to adalimumab in patients with Crohn's disease. I want to do a little bit of a look at the Varsity trial, the New England Journal study on the left, which was a head-to-head, double-blind, double-dummy comparator randomized trial conducted in 769 adults with moderate to severe UC that were randomized to receive either vedolizumab or adalimumab. The primary outcome was clinical remission at week 52. As you can see on this slide here, patients treated with vedolizumab had a greater frequency of achieving clinical remission compared to those treated with adalimumab. However, when you look at secondary outcomes, including corticosteroid-free remission, we actually see the opposite effect. And so my question for you is, are head-to-head trials like this the path forward? And if we look a little bit about that Varsity trial, it was a monumental undertaking. Hats off to the investigators who planned and conducted this trial in 245 centers across 34 different countries. I can only imagine how much this study could possibly have cost. And it was time intensive, taking over 4 years to complete. And that is efficient for a randomized controlled trial. There are a number of other issues. Dose escalation was not prohibited. And so I think we as practitioners need to ask ourselves, do these results really reflect real world practice? And finally, are these results generalizable to your study populations? I've shown some of the demographics of that study here. The median age, about 40 years, and 80% of participants were Caucasian. Is that what patients in your practices look like? And because of issues such as this and a number of other what we call the 6 S's or 6 shortcomings of randomized trials, many of us think that there's another complementary way to do comparative effectiveness research using real-world data and real-world evidence. And so RCTs are often described as being too small to detect rare outcomes or conduct clinically relevant subgroup analyses, too simple to detect interactions, too selected to be generalizable to all users and across all indications, too specific to assess all relevant outcomes, too short to detect long-term effects, and too slow and too expensive to answer all of the questions that clinicians and patients need answers to today. I'm going to delve into a couple of these concepts. The first is that patients in randomized trials often don't represent the real-world patients that we take care of because of the number of inclusion and exclusion criteria. This was a study here at Mount Sinai in New York, which looked at a practice of patients presenting to clinic and looked at the proportion of patients that were treated with anti-TNF who would have been trial ineligible. And in fact, a large proportion of patients that were treated with TNFs would have been excluded from trials from the reasons seen here. And what you can see on the graph to the right is that the proportion of patients who responded during everyday treatment was actually lower than those who would have been included in clinical trials, suggesting that the real-world effectiveness may actually be less than clinical trial efficacy. And this next slide here looks at the racial and ethnic distribution across 55 clinical trials. And importantly, only 10 studies reported race and ethnicity. And of these, the racial and ethnic characteristics are shown here: 85% white, only 3% Black, and 5% Hispanic. This is not only a problem in studies of inflammatory bowel disease, but across all clinical trials. They really don't reflect the demographics of the patients that we treat in our clinics. And so introduce this concept of real world evidence that can answer a number of different types of questions related to comparative effectiveness and safety, the real world use in special populations like children, the elderly, or pregnant women who would have been excluded from trials. Patients, we can study treatment strategies and treatment optimization methods. Often real world evidence studies use large and generalizable data sources so they can study long-term questions and a wide variety of outcomes, including cost and utilization. I'll start with just a couple of definitions. So real-world evidence is data regarding the use, benefits, or risks of a drug or device from sources other than traditional clinical trials. And what are those sources? They're often electronic health records, claims or billing activities, product or disease registries or multi-site studies, or patient-generated health data, including patient-reported outcomes, surveys, or nowadays, data from mobile health apps and devices. Real-world data really focuses on clinical effectiveness. And so what is effectiveness and how does that differ from efficacy, which is the end product of randomized controlled trials? And it's really an important thing to distinguish. So efficacy answers the question, does an intervention work under ideal conditions? Whereas effectiveness is, does the intervention work in the real world? And so the study populations of these two studies are quite different. Efficacy studies look at highly selected homogeneous populations, whereas effectiveness studies look at heterogeneous populations with few exclusion criteria. And comparative effectiveness takes this one step further, comparing the benefits and harms of alternative methods to prevent, diagnose, treat, or monitor a condition. A lot of real-world evidence studies use observational methods, including retrospective studies, prospective studies, in which case treatment allocation is not at random, and we often use fancy methods to control for confounding. And I'm going to give you a couple of examples of each. We'll talk about examples of effectiveness, safety, and research in special populations. And so starting off with effectiveness, this is a recently published study from the Victory Consortium, which is a multi-center retrospective study across 7 medical centers. It was a study that evaluated the outcomes of 212 patients with moderate to severe Crohn's and a median follow-up of 39 weeks. Looked at efficacy or 12-month cumulative clinical remission, as shown on the slide to the right. This outcome was achieved in about 35% of patients. And factors associated with not achieving remission are the prior use of anti-TNF agents, as shown to the right, and other factors such as smoking, perianal disease, and severity of disease. And so what about safety? This next study here was an extension of that consortium that compared vedolizumab to anti-TNF therapy. Primary outcomes were infections or non-infectious severe adverse events. And this is a study that used propensity scores to adjust for potential confounders. And so what are propensity scores? And forgive me for a minute as we geek out. I want to define a confounder as a variable that is associated with the exposure, associated with the outcome of interest, but not part of that causal pathway, or how the exposure might directly influence the outcome. A common concern in medical research is confounding by indication. that sicker patients are treated differently than milder ones. And so consider this example. You do a study and you show that ulcerative colitis patients treated with infliximab are more likely to have a venous thromboembolic event than patients with budesonide. Would you conclude that infliximab causes venous thromboembolisms? Probably not. In fact, you would immediately recognize that sicker patients are treated with infliximab, and sicker patients are more likely to experience thromboembolic events, and therefore disease severity is a confounder. There are a number of ways of controlling for confounders, the easiest being randomization, flip of a coin, and the other exposures should balance out evenly between the two groups. The common way, multivariable adjustment, create models so that when you look at the association between an exposure and an outcome, you take into account the potential for confounding by all of these other variables. Or the more complex way, which is propensity scores. And I'm going to try to explain that here. If there are too many potential confounders to adjust for in a traditional model, you can create a separate model to understand how potential confounders can be used to predict patients who were treated with drug A or drug B. And this prediction is called a propensity score. And you can then either match or weight the groups of patients treated with drug A or drug B by using this propensity score. And this can efficiently account for many variables all at the same time. Essentially, you remove the associations between the exposures or treatments and a whole basket of potential confounders so that you are then comparing apples to apples rather than apples to oranges. And so with that background in mind, and going back to the Victory Consortium here, they studied 1,266 patients, about half were treated with vedolizumab. And after propensity weighting, they found no major differences between anti-TNF and vedolizumab for most primary and secondary outcomes of safety or effectiveness. But if you look at treatment persistence shown at the right here, the probability of staying on treatment was higher for anti-TNF treated patients compared to vedolizumab treated patients. The next study I'd like to do here was actually done by our next speaker, Sid Singh. There were no head-to-head trials comparing individual TNF agents. And so Sid did a retrospective study using US claims data as well as propensity matching, and compared 3,205 new users of different anti-TNF agents with Crohn's disease. And so we already heard about propensity scores. What about studies with claims data? And so claims data are computerized encounter data that are submitted by providers to health plans. Claims data can come from pharmacies, hospitals, physicians. They make their way to the payer and ultimately can be licensed to a data user for research. Advantages include huge sample sizes, providing adequate power and precision, as well as the fact that these data arise from real-world settings. Disadvantages, however, include the fact that these data are not collected for research purposes and may lack a number of elements that you would wish to study. When you get these data, they look really complex with one line for each claim, and they really contain procedure codes and diagnosis codes shown in the middle of this slide here. But nonetheless, with a lot of art and science, an investigator like Sid can compare Crohn's disease hospitalizations and infliximab versus adalimumab treated patients shown to the left here, or hospitalized infections shown to the right here, and really learn about outcomes across large numbers of patients in the real world. I'm going to give you a couple examples now of special populations as well. The next study was a pediatric study comparing anti-TNFs to usual care, because at the top of this slide here, I'll remind you that the initial trials leading to pediatric approval of TNFs did not have control arms. And so we tried to replicate a study across a large network of IBD practices called the Improved Care Now network. which uses a electronic health records-based clinical registry shown here, where data collected at the point of care can be curated and used to calculate a number of outcomes, including disease activity indices. And as you can see here in this study, we compared outcomes of clinical remission and corticosteroid remission, corticosteroid-free remission amongst initiators and non-initiators of anti-TNFs, and found that these curves diverged around 6 weeks after the beginning of the, quote, trial, with an adjusted odds ratio of 1.5 and 1.7 for the various different outcomes, corresponding to numbers needed to treat in the range of about 5. And the last example I'll give you was a recently published example looking in the elderly, comparing anti-integrins to anti-TNFs. This is a study that used claims data as well as propensity scores. In this case, the claims data derived from Medicare because it was a study of the elderly. You all are now pros at those methods, and so I'll just go right to the data. 1,152 anti-TNF users, 480 vedolizumab users, and as you can see here, with the main outcome of infection-related hospitalization, patients who were treated with vedolizumab were less likely to experience this outcome compared to TNF-treated patients. And so there are a number of limitations of observational studies that we addressed over the course of this. Observational studies may lack data on exposures or outcomes of interest and may misclassify both the IBD itself or other outcomes. And importantly, despite fancy methods and propensity scores, can lead to residual confounding. And so in the last minute, I wanted to introduce you to this concept of pragmatic trials that can provide even more definitive data for high stakes treatment decisions. And so pragmatic trials are the latest rage because they really want to study the effectiveness, not the efficacy in routine practice settings. As evidenced in this review in the New England Journal, many efficacy studies can't adequately inform practice decisions because they weren't optimized for efficacy and therefore might overestimate the benefits and underestimate the harms. And so one example of a pragmatic study in IBD is a pediatric study where we are comparing low-dose methotrexate to placebo. Very few eligibility criteria, data collected during the process of routine care with very simple, easy to follow outcomes across a long follow-up period. And so with that, I'll conclude with a few take-home messages. Importantly, real-world evidence in IBD is really important and can fill the gaps that can't be addressed through randomized trials. As mentioned earlier, real-world evidence studies can be longer term with larger sample sizes, faster and less expensive with more generalizable populations, and can really look at special populations. And there's a number of advantages of observational studies versus trials. And in the end of the day, study design is a balancing act to find the methods to best address the research question that is important to you. Thank you very much.
Thank you, Michael. Last but certainly not least is Dr. Sid Singh, who is an assistant professor of medicine in the Division of Gastroenterology with an adjunct appointment in the Division of Biomedical Informatics at University of California, San Diego. He is also the director of the IBD Center at UCSD. His research focuses on comparative effectiveness and safety of therapies and management strategies in patients with IBD using network meta-analyses of clinical trials, observational comparative effectiveness research using large databases, and secondary analyses of clinical trials. He has led several IBD-related clinical guidelines developed by the AGA as a grade methodologist. His research is funded through PCORI, the NIH, the Crohn's and Colitis Foundation, AGA, ACG, as well as IOIBD. Over to you, Sid.
Thank you so much, Jessica, for that kind introduction. And thank you for all the wonderful speakers. It's truly been a great learning experience for me, listening to all the talks. I'll talk to you about network meta-analysis, which is another way of comparative efficacy of different medications and can be used to inform positioning of therapies. And like Jessica said, has been used more recently in guideline development. So these are the main objectives. We'll talk about what a network meta-analysis is, what are the key assumptions that go into it, and I'll walk you through a real-world example as to how we approached it for developing one of the clinical guidelines for ulcerative colitis. So we all know, and we've talked about this with prior speakers, and we recognize that there are multiple treatment options that exist for a particular condition. And in the field of IBD, head-to-head trials are still very rare. These trials are also expensive to conduct and take a long period of time, so the likelihood of future similar trials is also low. Most of the interventions, especially from a drug development perspective, are compared to placebo. And with that, it is difficult to recommend a clear winner or a medication that is better than all other medications due to lack of head-to-head comparisons. We're all familiar with traditional pairwise meta-analysis, wherein if drug 1 has been compared to drug 2, we can infer how drug 1 compares versus drug 2 based on the multiplicity of different trials. However, in the field of IBD, most of these trials are basically placebo-controlled trials, so traditional pairwise meta-analysis do not adequately inform the positioning or the comparative efficacy of these different medications. Network meta-analysis, on the other hand, allows for inferring a probabilistic relationship between these different medications, some of which may have been compared head-to-head, but a significant proportion may not have been compared head-to-head. As a result, a network meta-analysis develops this mixed effect, which combines evidence from direct head-to-head trials as well as indirect evidence through a common comparator. In this example, while we see most medications have been compared to drug 2, we do not have a way of inferring whether drug 1 may be more effective than drug 3. However, through a network meta-analysis with a common comparator in the form of drug 2, we can infer on a probabilistic relationship between drug 1 and drug 3 and assess whether one medication may be more effective than others, even in the absence of head-to-head comparisons. There are multiple different types of network meta-analysis, which are largely informed by this network connectivity. As you can see and imagine, most networks, until more recently in the field of IBD, were more of these star networks, wherein different agents had been compared to placebo. At the same time, as you can imagine, probably the best evidence is going to come from what we call a connected network, where multiple different agents have been compared with each other, even though the individual trials may have been small comparing head-to-head, but creating this connected network allows us to infer on the relationship and the comparative efficacy of these different medications much better. So what are the advantages of a network meta-analysis? First, it allows for a quantitative comparison of interventions that may not have been directly compared to each other, and this is pivotal for clinical guideline development, as we'll walk through one example. It combines direct as well as indirect evidence, which has the potential to increase the precision of estimates and often improves the certainty or quality of evidence in the GRADE framework. And finally, it allows us to simultaneously analyze all potential treatment options, so the entire network of evidence for a particular treatment, making full use of all the available evidence. And this allows for us to rank different treatment options and interpret how different medications may compare with each other and overall inform treatment approach for a particular condition. There are two key assumptions before we start going into a network meta-analysis example. Again, first is transitivity or similarity and comparability of trials. What does that mean? That basically means that the trials should be sufficiently similar in important clinical and methodological characteristics with similar distribution of key effect modifiers. In the case of IBD, disease severity at baseline, treatment dose, prior exposure to medications, the quality of these studies. Theoretically, patients enrolled in these studies could have been sampled from the same theoretical population. So if all of these trials were running simultaneously, the patient would have been eligible for all of these trials and would be followed in a similar way with similar outcome measures. This concept is called transitivity. The other key assumption is consistency or coherence of results. What this implies is in areas where there is both direct and indirect evidence available, there is consistency between those findings, i.e., a statistical agreement between the head to head trials as well as an indirect comparison for the same treatment comparison pair. These differences are believed to be nothing beyond heterogeneity. So, comparing it in the context of a traditional meta-analysis, a transitivity is akin to clinical or methodological heterogeneity for a traditional pairwise meta-analysis, and consistency or coherence is similar to the concept of statistical heterogeneity in pairwise meta-analysis. Now, let me walk you through an example of a network meta-analysis that we conducted for first and second line pharmacotherapies for moderate to severely active ulcerative colitis. If you're familiar with the field, there have been 15 randomized trials with close to around 3,700 biologic naive patients with moderate to severe ulcerative colitis. And this is the network geometry that we know of. And like Mike pointed out, Varsity was the one trial that added the head-to-head comparison between adalimumab and vedolizumab. Otherwise, prior to it, this was largely a star-shaped network. We recognized early on that prior exposure and failure of anti-TNF agent is a key effect modifier, and that's why we decided to a priori do two separate network meta-analyses within this study, first looking and focusing only on biologic naive patients, and secondly, looking at a subset of patients with prior anti-TNF exposure, and this was based on subgroup data that was presented in these trials. Again, recognize that most of these trials maintained the randomization because they stratified by prior exposure and non-exposure to anti-TNF agents. So all the key tenets of randomization still held across when we did this next set of network meta-analysis. And for this second line therapy, we focused only on presence or absence of prior anti-TNF exposure, identified 7 randomized trials or data sources with close to 1,600 patients. This is data presented in a graphical format, basically just showing if we put all of these different medications and their registrational trials next to each other, the bars in blue represent the placebo response across these different trials, whereas the different colors represent the efficacy or rates of induction of remission with biologic naive patients in the respective trials. And if we look at it like this, it seems like vedolizumab seems to stand out, but the placebo rates in this trial were also the highest. The general delta over placebo across most of these trials was roughly around 20%, at least for infliximab and vedolizumab, and somewhat lower for other medications. So let's walk through the concepts for network meta-analysis. The criteria for transitivity or comparability. Were there differences in enrolled participants in terms of clinical setting, disease spectrum, and baseline risk? For this study, we felt that all of these studies only included patients with moderate to severely active ulcerative colitis, which were defined in a very standard manner, and the baseline risks were very similar. Were there differences in interventions, the dose, duration, prior exposures, and how refractory the patients were? We made a conscious attempt focusing only on biologic naive patients when examining the first line therapies, and only looked at standard and approved doses of a medication. We did not include phase 2 studies, which tend to be overly optimistic for any intervention. Were there differences in background treatment and management, and was treatment evolving over time? We consciously chose not to include trials of combination therapy for this specific condition. Again, combination therapy meaning there was an intention to treat these patients and they were randomized to combining 2 different medications with monotherapy with either intervention. And finally, were there differences in the definition or measurement of outcomes? And we were pretty conscious in choosing consistent, well-defined outcomes across trials. There were subtle differences in the timing of these outcome assessments, varying from 6 weeks to 10 weeks. But overall, we felt that these differences were perhaps not too significant. And hence, we felt these trials were relatively comparable to each other and decided to proceed with a quantitative synthesis. I'll present the data on the induction of remission and mucosal healing or endoscopic improvement. Here, this is a typical way of presenting data for a network meta-analysis. The top line or the blue section presents data for comparative efficacy of different interventions for induction of clinical remission, whereas the green section represents the data for a different outcome, which was induction of endoscopic improvement. The bold and the dark blue represents the results that were statistically significant. And as expected, all of these interventions were more effective than placebo. If we look at different agents, the one treatment that stood out, the one comparison that stood out was infliximab was more effective than adalimumab in inducing clinical remission. And when we look at endoscopic improvement data, similarly, we observed that infliximab was more effective than adalimumab or golimumab for induction of endoscopic improvement. Another way to look at this data, like I said, was ranking or the probability of these different agents. If we think of a perfect agent as a rank 1 with a Sukra score of 1.00, we rank these different agents through sort of a Markov modeling kind of a process, and inferred that infliximab had the highest probability of ranked highest for induction of clinical remission, followed by vedolizumab. Again, important to keep in mind that Sukra or ranking probabilities by themselves are not the end goal here. And there is no statistical comparison between Sukra rankings, but rather you're inferring all of this data and putting it contextually with other data sources, like Mike and others alluded to, from observational data sets when making these inferences. A way to present this data was how does it compare for absolute rates of achieving induction of remission and endoscopic improvement. With an illustrative example of placebo rates of 10% for achieving clinical remission and adding in the odds ratios for these other interventions, we observed the corresponding rates for induction of clinical remission of 30% with infliximab and 22% with vedolizumab, and corresponding rates of endoscopic improvement in excess of 50% with these two agents. We used grade criteria to a certain quality of evidence. What about second line therapy? Here again, going through the same framework, were there differences in enrolled participants? And it was important to figure out and say that while we combined and lumped all patients with prior exposure to anti-TNF agents, we were not able to differentiate whether or not these were primary non-responders and inherently severe group of patients versus whether these patients had secondary non-response or intolerance to anti-TNF agents, patients who may not be as intrinsically treatment refractory. And similarly, in terms of background treatment, we could not differentiate patients who had failed a single anti-TNF agent versus multiple anti-TNF failures, the latter potentially representing a more difficult to treat patient population. So the data from this needs to be interpreted with some caution. Here are the results, and here what we observed is that ustekinumab and tofacitinib seem to be more efficacious as compared to vedolizumab and adalimumab, both for induction of clinical remission and endoscopic improvement. Keeping in mind the concept of prior exposures and multiple exposures, we felt that since trials of tofacitinib and ustekinumab were more contemporary, they probably included patients who were more treatment refractory, having failed multiple prior therapies, as compared to trials of vedolizumab and adalimumab. And this is similar findings for the Sukra ranking. How did we use these data? How about maintenance of therapy? Again, we felt that there were differences in trial design which precluded this indirect comparison. We know that trials of infliximab were primarily treat straight through trials, whereas trials of golimumab, vedolizumab, and tofacitinib re-randomized responders to induction agents, and we did not feel that we could differentiate or combine these two trials of maintenance therapy. So overall, this is great for induction of remission, not as good for maintenance of remission. We use this data with Jessica to develop the moderate to severe guidelines for ulcerative colitis for the AGA, and use this data to inform specific aspects of positioning of therapies. In biologic naive first-line therapy, the AGA suggested using infliximab or vedolizumab rather than adalimumab, leading to a conditional recommendations suggesting the use of infliximab or vedolizumab rather than adalimumab. Of course, there are aspects related to patients' values and preferences that go into any conditional recommendation. And similarly, for second-line agents in patients with prior failure, the AGA used this data to infer that in patients with moderate to severe UC who have previously been exposed to infliximab, particularly a subset of refractory patients with primary non-response, the AGA suggests using ustekinumab or tofacitinib rather than vedolizumab or adalimumab. What are key issues in interpreting these network meta-analyses? First is an inability to account for important disease modifiers at the study level, most importantly, prior anti-TNF exposure or prior treatment refractoriness. There is an inability to account for differences in trial design, which adds on additional layers of assumption, which is why we were not confident in doing a network meta-analysis of all maintenance trials together. There is a problem with inclusion of only phase 2 studies of experimental therapies, since these are smaller trials with low event rate and introduce instability in network by being overly optimistic with all phase 2 studies. And oftentimes there is over-reliance on ranking probabilities or SUCRA, and there is clearly a lack of critical appraisal of the quality of evidence or the confidence in these estimates in making appropriate treatment decisions. With that, I'll summarize that well-conducted network meta-analysis can inform comparative efficacy of different therapies and provide robust evidence for clinical guidelines. There are key assumptions of transitivity or comparability of different studies, as well as coherence and consistency. A well-connected network is likely to result in more stable estimates and more reliable estimates. Finally, the concept of a living network meta-analysis, which is updated with new trials, will iteratively and comprehensively inform the entire body of evidence. And this is very evident from the COVID NMA initiative, which is a living mapping and a systematic review of all COVID-19 trials. With that, I will stop, and I'll be happy to take questions, moving it back over to Jessica.
Wonderful. Well, what a series of excellent talks, and we've been getting some great questions from the audience. So now that we're all together, why don't we start with Martin? Martin, we had some questions about how do we think about using external control arms if the endpoint definitions or standards have evolved since the time of that initial comparator study? How do you think about that?
It's all about fair comparison, basically. So if your trial is using the novel definition, then probably that historical control would be an issue. And you don't want to go too far back in time, because at the end, you really want to compare the two sets of outcomes. So probably you would maybe need a prospective study. But again, it all depends. Question number one is, why are you doing the external control arm? Is it to benchmark your product? Is it a head-to-head comparison? But again, depending on all of those questions, you would probably need to adapt and select the patient that would fit the novel definition.
Great. And over to you, Niels. Could you please clarify what further insights you've had post real world evidence versus information gathered during clinical trials in the development of Entyvio?
Sure, yeah. I think this is probably something that is applicable to all drugs. When you develop a population PK model just on clinical trial data, you're working with a selected cohort of patients. For example, there may be restrictions on body weight for patients that are being enrolled into the trial, as well as severe disease characteristics. So what we're seeing in the real world is that you see greater variability in those different covariates. And so what eRelay did for us for vedolizumab is it gave us an opportunity to see if the originally developed population PK model from the trials was able to fit the actual data observed in the real world, so to speak, in clinical practice setting, where some of the covariates of individual patients may have gone beyond the typical range that we saw in clinical trials. So we were able to confirm that the model indeed was able to fit the data. As well, by using that approach, we were able to then look at the exposure-response relationship. So using that approach allowed us to go beyond trough concentrations, which what has been typically reported. So our novel findings are more around clearance. That seems to be the strongest predictor, stronger than any exposure variable, to be predictive of long-term outcomes. But we also found other predictors of great importance, such as prior IBD surgery, the age at which vedolizumab is started, and others as well. So I think collectively that gave us a good grasp on better characterizing the exposure-response relationship and accounting for some of the variability that we observe in clinical practice.
Great. And so, I mean, we're talking about real-world evidence and this modeling because it seems that clinical trials, especially in adults, are just so difficult to recruit and enroll in. How do we think we can fix this problem, or can we fix this problem? Michael, what do you think?
Yeah, so adults are easier than kids. Let me start with that. But I think there's two sets of complementary issues going on. One are protocol-related issues, and the other are site-related factors. In terms of protocol, we see that most of the trials to date have had really strict eligibility criteria, a whole list of inclusion and exclusion criteria. Like I showed you in the Mount Sinai study, many patients who are good candidates for a treatment after approval would actually have been excluded based on the trial protocol. I think that many of the trial protocols are quite intensive and involve repeated visits and repeated colonoscopies, which are not appealing to many patients. We have more treatment options than ever before, which means less patients who are waiting or wanting to try experimental medications. I think the long washout periods are problematic and makes it really hard to get patients who want to be in trials and might be eligible for trials actually to be able to safely enroll without getting sicker during the washout period. And then I think there's a number of site-related factors as well. Many sites are optimized to provide clinical care and to provide clinical care at a fast pace. And clinical research is an afterthought rather than the primary objective at many trial sites. And increasingly, there's competition with a lot of clinical trials all looking for the same types of patients. And so accrual of patients into any single trial will suffer a bit with more and more trial choices or opportunities.
And I think we just have time for one more question. So I'm going to start with Martin. Would you consider using historical external control arm safety data for diseases, such as RA or MS, for early phase safety trials in IBD, or are these populations just too heterogeneous and we can't compare?
Well, you can use external control for safety, but what you want is a stable standard of care. So I think the questions could be answered in two different ways. If you're thinking about using your external control arm from a disease population, so a population that have RA, MS, and so on, I'll be careful with that because even though they're immune diseases, they tend to have higher comorbidity. So, and it all goes back to why do you want to perform that? If it's a head-to-head or a benchmark analysis, I don't think having an heterogeneous population would be that harmful if you want to benchmark your product, because that way you're playing a bit more conservative. But at the end, it's really about balancing the comorbidity between the different cohorts.
Guys, what an excellent discussion today. If we did not get to your question, we sincerely apologize, but we will be happy to answer it offline. So just provide us with your email address. And I will turn it back over to Vip for closing remarks.
Great, thanks so much, Jessica, and thank you to you for chairing the event, and to all the speakers, Martin, Niels, Mike and Sid. That was a real phenomenal tour de force of where we are today in IBD with RWE. You know, I think many of us really think of this mainly in the comparative effectiveness space and post-marketing space, but I think some of the things we saw today, particularly from Niels, show how this might be incorporated earlier on through the drug development life cycle. And I think with the regulatory guidance allowing real-world evidence to be submitted, I think in the coming years we're going to learn a lot of what that, and you know, what kind of rigor we need to produce that data with. So again, just thank you to everybody who attended and spent longer than 1 hour with us today. Really appreciate your time. And our next event will be sometime in February or March of next year, and there'll be more details to follow about the topic. So again, thank you to the whole panel, and thank you to everyone for attending. Have a good rest of your day.